Keep it going now.
Okay, great. So I will talk about scaffolding and how to confirm whether the scaffolding step worked or at least did something, and how can we check that, and how we can share that. So going back to the pipeline, um, this was the overall overview of the pipeline, and today I'm gonna talk about these steps, mainly how we go through Purge Haplotix, Scaf10x, Bionano scaffolding, and Hi-C scaffolding, and then the first polishing step using the Arrow pipeline. When you go to the VGP Tools project on DNA Nexus, actually I can pull that slide, no, pull that web page. Okay, I got logged out. All right. So when you go to VGP Tools, you can see each scaffolding steps are made by courtesy of Maria and Chai. So except for the VGP FileCone and unzip workflow, all the rest of the steps are made as scaffold and step number workflows. So you copy from here to your project and then put in the inputs. Go over on details for each step. So for Purge Haplotix, there are two main inputs when you click the app. The first one is the input FASTA. The second one are the subreads, file reads. Purge Haplotix is aligning the PacBio reads back to the FileCone unzip primary context, the C1 FASTA file, and get the read depth distribution here, and then prints the histogram image as well as a text file containing the genome coverage. So here it takes a little bit of manual effort to figure out for the next part 2 app. We need to get a few things. The first thing is we have to identify the haploid peak and the diploid peak. So the diploid peak, if you get only one peak, sometimes it's a bit tricky to figure out whether that is a haploid peak or a diploid peak. Usually, if the genome is very low in heterozygosity, you will see only one peak, which normally is almost the diploid peak. And the diploid peak here, the coverage, here it says about 70x, should be almost equivalent to the estimated sequencing coverage that you can get from the total number of bases in PacBio reads. This is an output from when you run FileCone. At the very beginning, there is a page. When you scroll down, you can see the FileCone outputs. Here you can get the total number of bases and divide that simply by the estimated genome size. It's mostly the haploid genome size, so it's the same one. You can put the same one as you started FileCone unzip, or you can put in the GenomeScope estimated haploid genome size. But it should be almost identical to the diploid peak sequencing depth. So what we want to do here is to identify contexts that have the haploid peak coverage and find the pair and get rid of one of it. So we need to define the boundaries of these peaks. And while doing this, I would encourage you also to add the genome size to the VGP project tracking sheet, because we are going to use that later on. I will come back to this later in my slides. So once you have a sense of approximate where to pick the boundaries, we can proceed to the next part 2 app. Here you can see it needs the low cutoff, midpoint, and the high cutoff. And I'm not sure what the alignment coverage here is, but I assume this should be the diploid peak. And change the junk threshold to 200 and suspect threshold to 0, so we don't accidentally throw things away.
Arang, if you want to, I think we can change that to default value from here, so prevent user error.
Yes, I think that will be nice. It might be a bit tricky for the diploid peak, though. I'm not sure where this comes from. I don't think this is a parameter usually provided to per Chaplotix. So while doing this, can you check for this?
Yep, sure.
Thanks, Chai. And so what are the low and mid and high points? Here, this is the low point, this is the midpoint, and for the high point, I picked the same coverage where it meets the beginning, like the first dropping point. And this is a very simple script that I wrote to automatically determine the LMH boundaries. You can copy this and run it locally by downloading this GeneCop file, which is the text file from the part 1 output. And this jar file is available on our GitHub here. So once you got the LMH, compare that back to the plot and see if that matches it. My script almost works for every cases, but it might get a bit in trouble if there's only one peak available. If you see any of those kind of behaviors, please report back to our training channel. So at the end, you get 2 FASTA files plus some other output files. You will get 1 curated FASTA file and a curated Haplotix FASTA file. So this curated FASTA file will become the P1, and the curated Haplotix will become P2. And I would encourage to get the assembly statistics, which I will cover later on, and compare that to the C1 stats and see if there are any changes. Usually, if you run per Chaplotix, because you get rid of some small contigs, you can see that the N50 sizes or the NG50 sizes goes up. And the total number of bases will also slightly reduced because some of them got dumped into the Haplotix. And just briefly check if you see any abnormal names in your FASTA file. I believe most of them are now covered and configured in the DNA Nexus app itself. And you need to concatenate these C2 and P2 later on to Q2. I saw for one genome, I think MyHeat used the file concatenator, but I just talked to Chai, and he was planning to make the Salsa app available to accept these 2 files directly, so we don't need to create an additional Q2 file. Excuse me. Yeah. And the last step is export everything back to our GenomeArk. Okay. Do you have any questions regarding purge haplotics? Anytime if you have.
Well,
Yep.
I may have one. So,
Mhm.
so purge haplotics essentially works in terms of read depth, right? So it's separating. And what happens in the middle? So what happens where you have, let's say, the overlaps of the two distributions?
Overlaps of it. If you have just one peak, you mean?
No, I'm wondering, I mean,
Mhm.
how the script works. Because when we generated the haploid genome for the bound swallow, we used similarity search instead of read depth to separate the alleles, and we had two clearly distinguished peaks. But here, for instance, in this example, you have overlapping peaks. So I was just wondering what happens in the region of overlap, if you know.
It runs internally Minimap 2, the latest purge haplotype. It used to run, what was it, last Z inside, but then it switched to Minimap 2 to identify, using the context having the haploid peak coverage as a query. It aligns back to the rest of the context to find the pairing haploid type.
Okay.
And then it gets rid of the smaller one.
So it's essentially a way to identify the contexts that are putatively unique in the first place.
Yes, I think so, yes.
Okay. Thank you.
So yeah, it remains the one, whatever was longer and was including the other haplotype. So the shorter one will get dumped into the purged haplotype.
I see. Okay.
And okay, let's go to SCAF10X. We had quite a bit of optimization going on this particular step. So here again, we use the purged primary context as the assembly template that we're gonna scaffold. And this is the read 1 and this is the read 2 of the 10X reads. It's very simple, and internally it runs 2 rounds of scaffolding. So you will see only one final FASTA file, but it's already gone through 2 rounds of SCAF10X process. So at the end, you expect to see one scaffold FASTA file and an AGP file. The FASTA file will become the S1, and you can check the AGP file to see whether it actually scaffolded something or not. In the AGP file, there is the scaffold ID and all the contigs that got joined into that scaffold. So you can check that AGP file quickly to see whether SCAF10X actually did scaffolded something or not. And we also want to see the assembly statistical changes, so get the assembly stats. And here there is not so much to modify. The scaffold names will have a prefix of scaffold already, and will be numbered. So there's not so much to change here, then export back to the GenomeArk.
Arang, what is the get assembly stat that you looking forward here? Is it calculate this distribution or something more than that?
It's simply measuring the size.
Okay, then I think I can add to it.
And this is the script that Maria was asking Julia to make an app for this.
Oh, I see.
Yeah, so I think until we have the app on DNA Nexus, you can just run it locally. It's a very simple script.
Okay, I see.
Yeah.
Well,
I want
maybe.
to,
Yeah. Sorry. Go ahead.
sorry, I want to add one detail here for this Captain X, you on the slide. So, would you mind if I share my screen for a second?
Yep.
Um, I'm sorry, I'm not very familiar with X. How do I share my screen, by the way?
When you move your mouse down, you can see an arrow that's pointing up. Click that. Yeah.
Is it working now?
Yeah, I can
Yep,
see.
sounds good. So, first of all, you all might see in Arang slide that there is a recommend, the way that we can name the 10x input here.
Mm-hmm.
So, I just want to mention that sometimes the data that got uploaded long time ago, they didn't have the same, uh, kind of naming here. And if you click on this app, try to find the input, you can see here that there are some suggested input that already, have the matching name pattern here. But I hope that it not going to happen again, but there was one case in the past that, there are some input that match the name pattern and some input that doesn't match the name pattern because it have just, species name and then just say underscore R1 and then I think it doesn't have 001 here something. So I recommend that if you guys want to do sanity check, you can go to this 10x folder and you can clear the pattern here. So don't be panicked that you see a lot more file here because this is like R1 and R2 now it all show up because when you clear the pattern it tries to find all the file here. But I just mentioned that it is a good sanity check, try to come to this and then try to select the try to count whether you have like the right number of the input here. Say this is 5 and that's actually the same as what you see earlier. So even that you're good. But sometimes there might be more input here. So make sure that you come and take a look whether you actually try to use all the input that we have here. Yeah, that's all I want to share.
Yeah, thanks, Chai. That was very important. Okay.
So here, just to make sure that I understood, so here you had 10 files of sequencing run and 1 index files, right? But you want to select only the forward reads.
The forward, yes. The reason I suggest you guys try to clear the filter and take a look at the folder yourself is because sometimes data come from different provider, and then some provider they didn't have the naming convention that 10x uses as a standard. So I already communicate with Oliver that going forward we would like all the input to be renamed according to the standard because later on when you run the long ranger, it gonna require that pattern anyway, otherwise the app gonna crash. But
Right.
meanwhile, for the next month or two, data that you got from Oliver long ago, they might not have this name combination. So make sure that you check first.
Yeah, if you see any differences in what you expect and what should be there, what is there, please report back because that was our file naming convention. It should follow all the files on GenomeArk, especially the genomic data, has a specific file name convention. If you see any malformed files, please report back so we can fix those as well. Yeah, and it's important to note that the Lone Ranger does not accept a malformed 10x read. Thanks, Chai, for mentioning that.
Yeah, it will crash explicitly. It basically don't know that this file exists. So
Right.
And Chai, you had, I think you said something about the stats that you could add something there, or
I think we have the app that can calculate like risk and distribution, those kind of things. But I would like to hear more from Arang after the scaffolding, like what are the stats that she has in her script because she might have more stuff there, which I suspect. So yeah.
so maybe we can next week we're supposed to discuss this app as well, I think. So maybe we can look into it a bit further.
Yeah, we can sing at a time.
Okay. Thank you.
Okay, I guess we can go over to Bionano. So there are some genomes which were done with DLE1. I'm not sure if it's correct to say that it was sequenced because it wasn't really sequenced. But anyway, we have some genomes that only uses one enzyme and some genomes that uses 2 enzymes. So if your genome, if you go look under the genomic data Bionano and you see DLE1, then you will probably need only the one enzyme pipeline. If you see BSPQ1 and BSSF1, you will probably need the two enzyme pipeline. So the one enzyme pipeline, again, takes the scaffolding, the primary scaffolds that we got from SCAF10X, and the ZMAPs. Here, this ZMAP will probably have DLE1 in the name. It's again very simple. It runs through this Bionano hybrid scaffolding app and does file concatenation for the NCBI version of the final scaffolded FASTA file and the unscaffolded FASTA file. And then it runs through James's N trimming script, which detects large stretches of ends at the beginning and at the end of a scaffold, and also the small bits of the enzyme, the DLE1 sequences that got inserted from the CMAP to FASTA conversion, and gets rid of those. And it's also useful to see the log that this step produces. It's this step, I think. It prints a warning in the output list for those scaffolds that have more than, I think, 70 or 80% of N bases in the scaffold. Yeah, and the last step here is, I think, provided from DNA Nexus that simply gives you a few statistics of your scaffold result.
Around here, could you recap the reason why there are so many ends to be trimmed?
I think it was a bug in the previous Bionano hybrid scaffolding app. So when you have CMAPs that go longer than the contigs, let's say if you have a contig here, and I think I need something. Okay. Let's say if you have a contig and you have CMAPs that go across, like at the end, over the end. It has a few enzyme sites at the end, and those will get transferred to bases, 6 Cotter bases, I think, and the rest will be just placed at ends.
I see. So it's not really a bug to some extent. I mean, it's somehow telling you the length of the DNA fragment.
Could be, but by design, the NCBI version is getting rid of those.
Yeah, I see.
Yeah. So I think it was a bug that the Bionano app, Bionano software had.
Okay.
Yeah. Right. Any more questions? Okay. And this is the Bionano 2 enzyme workflow. Again, it takes the same FASTA file here and 2 enzymes here. You have to set the enzyme name here, and these should follow the order given as in the input. So if you put in here the BSPQI, you should set this to BSPQI. But I don't think the genomes that got recently submitted have these 2 enzymes. I think most of them are now routinely done with DLE1. So at the end, again, we get the scaffolds, the concatenated scaffolds. which should then be renamed to S2. And again, Bionano outputs an AGP file as an intermediate file. So check that again and get the assembly stats. And make sure to remove all these semicolons because these will cause troubles in Salsa, which is the next step. And don't forget to export any files at the end. Okay, any questions?
Regarding the colon here, is that already fixed by Maria? I think she put in some custom or command to fix it inside the app, right?
Right. Yeah, Maria already fixed that internally in the app, but I know there are some genomes that ran the earlier Bionano scaffolding.
I see.
Version. Yeah. And okay, this is the last scaffolding step, the Salsa app. Here you put again the S2 FASTA file, and under genomic data there are either ARIMA, Dovetail, or FACE. I think most of the genomes that you get assigned have ARIMA Hi-C data, but some of the genomes that got sequenced earlier have either Dovetail or FACE genomics. Oh, I forgot one thing that I wanted to tell. Let's see if I can pull this out. All right. So when you go to Salsa app, there is one thing that is required here, the restriction enzyme basis. Let me just make sure that so for ARIMA, it's GATC and GANTC. I will modify this. I will add that later in the slide. But it's GATC, GANTC. And for Dovetail and FACE, it's just GATC. So take that in mind and just run it. It's doing the indexing, the reference, and maps the Hi-C reads each separately. Here it accepts the forward reads and reverse reads. So here goes the R1 and the R2 reads. And it maps the reads independently because BWA normally assumes the two paired ends to be aligned within a certain length, within a certain distance. So if it sees a read 1 and read 2 being aligned too far apart, then it thinks that this is a bogus alignment and throws it away. So we want to prevent that happening, because that's usually what you see from the Hi-C long interactions. So the ARIMA pipeline is aligning the reads each independently and then trims off the 5' end of when you have a chimeric read with the cotter in the middle of a read. And here at the end, this was inserted recently just for the BGP pipeline because we need to concatenate the alternate haplotics as well for the next polishing step. This step will be changed, I think. So here it accepts the Q2 file, but later on it will be also accepting two files, which will be the C2 and the P2 file, the original FICON alternate haplotics, and the purged haplotics, purged haplotics, which was the Q2. And again, the output here will be the S3 file. This is the SALSA output, and you will get one more output, the S4, which contains both haplotypes. So when you get the assembly statistics, you will see that from S4, the number of bases will be almost doubling the genome size. Not always, but depending on how much Vicon and zip worked. And don't forget to export to GenomeArk. And this is...
Can I ask something?
Sure.
I have some question on input of SALSA.
Okay. Yes.
After finishing the BioNano, then I got the FASTA file and trimmed FASTA file. What is the difference between both files? And how can I use how can I choose for the input as an input?
Yeah, the trimmed FASTA file is the one that is produced by the BioNano. Oops, where is it? Here. By this step. So I would say use the trimmed FASTA file to prevent any artifacts. Does that make sense?
So do you recommend to use the trimmed FASTA?
Yes.
Yeah, rather than FASTA file. Okay, thank you.
Yeah. So please rename the trimmed FASTA file to S2, and then use that for SALSA. Okay, are there any other questions? Or can we proceed? All right, so the next step is the arrow polishing step. It has a very long name. I think it's following the PacBio tools name, which is the BAM sequencing workflow. Here, again, this will become the subread.bam, and this will become the S4. And make sure that we have both haplotypes in this S4. So we are aligning to the whole assembly instead of just one haplotype. It's very simple. It aligns back the subreads using Minimap2, and then does run the arrow pipeline here. So at the end, you will get a polished S4 file, and rename that to T1. And again, get the assembly stats and export this to GenomeArk. Here, the names will have a pipe arrow at the end of the name. And I forgot which step it was, maybe Chai can chime in, that we replace all the pipes back to underscores. I think that was before running Long Ranger.
That I think is let me check.
Yeah, but Freebase throws out some warnings if it sees the pipe. I don't think if that was because of the pipes, but we're suspecting that this is the case. And I think that's all for the workflow. Does anyone have a question so far?
So we want to put the Freebase one on hold right now, right?
Right. So I'm checking the Freebase. I checked the Freebase QV that I run on the Hummingbird, and I saw that the QV was almost like not changed.
I see.
Yeah,
Good. I think it's good to investigate first.
yeah,
Uh, I have one pretty small detail actually, but at the BioNano step. So
yeah.
I saw that there are some other research group that they provide the specific XML file for the configuration, and I heard from them that if they use the configuration file that's specific for that enzyme, they got slightly different number of it, and they mentioned that that's because the p-value cutoff that provide that configuration match with the enzyme better. So I just wonder whether like BGP group would be interested to incorporate that into the pipeline as well. If so, I can provide more detail after the meeting today.
Yeah, I think I like that option too. It's the high C option, which tolerates more alignments when aligning the C maps to the, I mean, between the C maps, which tolerates more errors basically for extending the mapping. I like that option too, and I think the VGP, uh, at least those who are running the BioNano step locally, started to use that option. But I saw for some genome step that option didn't work.
Huh.
So I think we have to make an option to try that first and then go back in case it fails.
When you mentioned that option in the SALSA, is it at the ARIMA or at the SALSA step?
I mean, running the BioNano hybrid scaffolding.
Oh, I'm sorry. I confused because I heard you said high C. Okay.
Yeah, it's confused because the option, the XML file contains high C in the name.
I see. I see.
Yeah.
Uh, I see.
It was specifically designed to error correct the high C inversion errors that you can frequently see when you have small contigs ran through high C scaffolding without any other scaffolding steps.
Yeah, but the high C was run afterward, right? So
Right.
that actually is not applicable in our pipeline, is it?
Yeah, the only benefit I think will be that it will better detect inversion errors that were placed either by Falcon or Scaf10x.
I see.
Mhm. Yeah.
I have a general question here. So, apparently, at every step of scaffolding, you are required to rename scaffolds. Is there a specific reason for that? Isn't that possible to make it automated? Because I suspect, I mean, that could generate some problems for, I mean, to say a more general public to if they want to work on it.
Yeah, that's a good idea. I think we can
Okay.
do that.
I would say that technically, yes, we can rename everything according to the flow convention. But the thing is that we also use this pipeline for other customers as well, and it's gonna be. I just don't want to run into the conflict that someone else use the tool that we maintain on the platform here, and then they got whatever information that we can put together up there.
Mhm.
So it I think I need to talk with Marie and
Okay.
investigate whether it's okay because you guys have embargo policy for both knowledge and both big data, right? So
Okay.
Yeah. Maybe if you get trained how to make a DNA Nexus app, you can change the output in the workflow.
Right. Okay.
Or add another app that changes. But
Yeah,
I
I mean,
think,
that will be easy, I think. Okay.
yeah, but I agree with Chai that because the apps are also used in other workflows outside of BGP, and those apps, I mean, the very small units in the workflow, in each workflow, have their own, are following the original tool's naming convention. So I think it's okay to rename it manually, but to prevent will be also nice if we cannot.
Yeah, I mean, if there is like, say, one accidentally introduces a typo and doesn't realize what's going on, so I mean, it can produce some noise.
I would say that all the custom stuff, say trimming and concatenation those things. So I can tweak it to output the correct name to begin with. But
Yeah.
the data that come out directly off the standard app, that one I prefer not to modify it because it's gonna be odd, like somehow other customer got like S4 out of the
Yeah, it will be a bit awkward.
It will be a way to promote the VGP outside the VGP community.
Just a quick question around, do you plan to do you have more training material today that we can talk about, like, how we're gonna inspect the HEP or how we're gonna inspect the high C or bio-nano result?
So
Oh, okay.
I have, yeah, I think I don't have the slides of an high C example, but I can add that maybe at the end. So I will go over of the how I got the assembly steps and use that as a sanity check for each step. So when you go to the VGP project tracking sheet, you can see that here you can see all the genome identifier and and here is who is currently running this genome. And the important things for sanity checking I use are these columns here. So you put in the genome size here. Here, preferably, I would ask you to insert the one that you got from GenomeScope, so we don't have to go back and dig into the files to find what the genome size is. And for these columns here, these are the N50 values of each step, not all steps, almost all steps. And these are, when you switch to other tabs here, you might have noticed that there are the abbreviations with an N here at the end. So this here is again the identifier, and from here these are the total number of bases, total number of either contigs or scaffolds, the maximum size of the unit, and the N values here. So you can see, let's say, for example, here, this is Marcella's sloth genome. You can see the maximum contig size was 57 megabases, and the N50 value, I see that something went wrong here, so it's something in between these two values, I think. Yeah, so it's around 8 meg or 9 meg, I think. So the way I pull it out is just a simple VLOOKUP that's provided in Excel. It looks for the genome ID, and this column places the value of this column, the N50 column, back into this sheet. So you don't have to touch this side. Just insert whatever you got into the particular sheet. And how do we get this stat is locally you could download the FASTA file, unzip it, and run the ASM stats script. It's a very simple script. It produces a length file first to get all the FASTA entry name and the length, and the non-N base is length. And from that, internally, it sorts by the length and sums up all of them and picks up the length that from that covers the half of the assembled size when you sum up from the longest one. And the script also prints out the NG values. For doing that, instead of using the full genome size, the full assembled genome size, it uses the given genome size as a denominator. So here, for example, if my genome was 1.1 gig, you give 1.1 gig base as the genome size and the FASTA file. And for those which are contigs, already contigs, place a C at the end and run it. Then you will get the assembly stat just straight from the assembly itself. If you don't place anything, it gives you plus the contig stats and the gap stats. It's not generating a separate FASTA file for getting the contigs. It just grabs out the gap positions and does a subtraction of the original length and calculates the N50s and the NG50s from that. And it also produces the gap statistics, like the N values for the gaps. It is useful to check whether the scaffolding step introduced some kind of large N-based gaps. And from here, the last step here, this P3 is the one that went through 2 rounds of long ranger polishing. Here at this point, we want to split back the FASTA file and give it back to Kirsten's group for curation. So I just simply made a P option, which does all of these above, and plus it gives you the primary stats and the alternate stats, and splits the primary and alternate into 2 different FASTA files. I will probably come back for this in the next training session. Once we have the Long Ranger FreeBase polishing app available, I will go over. I think it's a good timing to go over for both of them and direct you how to submit genomes to Kirsten's group. So here I noted again the checkpoints. Look for any large gaps, look for the assembly total size, and look for the N50s and so on. Here again, in detail, just as a simple sanity check, the trend should be, the N50 trend should be always increasing. Oh, there should be another arrow, sorry. From C1 to P1, P1 to S1, S1 to S2, S2 to S3. This is because we get rid of small haplotics. This slightly helps increasing the N50. And from here, if you do the scaffolding, you expect that the scaffolding step did something, so the N50 should also increase. But if you don't see much changes in these S1 to S2, S2 to S3 steps, you can always go back to the full spreadsheet, check how the N1 to N50 values change, like the maximum and the N10 and so on. Sometimes only a few changes might happen and might not affect the N50. And also again, I put a comma in here because from here, T1 contains both the primary and the alt. So the N50 drops. Don't panic if you see that. The N50 drops because we are now including all the small alternate haplotics in here, which will affect the N50 values. And again, from T1 to T2, T2 to T3, you can see the N50 values are not like dramatically changing, as you can see from here. It's only T1 and T2. From this step, it's mostly affecting the small indels that get polished. So you wouldn't expect to see that too much changes in here. Okay, and I think this is my last slide, almost. Yeah. So a bit more complicated sanity check. At the end of scaffolding, you can run Juicer and Juicebox with the Hi-C reads. And I'm also trying to figure out how to run. There is a way to convert the Salsa intermediate file directly into an Hi-C file that you can import to Juicebox to visualize the interaction as a heat map. I will probably go over that again once I have a clear understanding of that step. And at the end of polishing step, you can do this for each polishing step too. You can check the VCF file. I know that Arrow and Long Ranger Freebase both produces VCF files containing the changes that got applied. And also from the BEM file, you can collect the bases that have certain amount of minimum alignment. And from that, you can calculate the QV. You can also run Busco to see if you get any increase in the Busco genes. Usually, if the QV value goes higher, when the base pair quality goes higher, Busco gets easier to detect the genes that were missed before. Doesn't mean that the Busco gene wasn't originally in the assembly. It was there. It just had too much error that it wasn't detected as a proper gene. So you can see from Busco that either the fragmented Busco genes gets reduced, or you can see an increase in the complete Busco genes. And before sending for curation, you can do these steps locally and send it along with the assembly to Kirsten. But I think it's better to cover about this in another training session once we have the assemblies ready for curation. Okay. So I can show you some Hi-C heat maps and talk about that. Before doing that, do we have any other questions?
So right now we want to hold off data that we want to send to Christine, right? Because I think we have a separate chat channel that we said that we're gonna send a lot of data to her. Maybe we should notify her that this data has some problem at the Long Ranger step, so we want to like hold on that first.
Right.
Okay.
So I mentioned that on the assembly channel, I think.
Okay.
To hold it off. So I'm rerunning the one more QV step, Long Ranger Freebase step locally.
I see.
My pipeline prints at the end the QV of the assembly that was given, like the input assembly QV.
Mm-hmm.
So I can check what the QV is from the Illumina data.
And also, if you already have the script that can calculate the QV here, I think I can make the app for that. So it should be very helpful for us to check this like upfront. Without any concern that we wait until we get all the assembly result and check this
Hmm.
by waste of money. So
Yeah, I think that's a good idea. Let me pull up the GitHub page. So
don't worry, you can send to me afterward. The just the location of the pipeline.
yeah,
I
so
see.
most of the scripts that I'm running
Oh,
locally are here. There is a QV separately,
uh-huh.
but my Freebase Polish pipeline uses. Let me see. Let me check. So here it runs Freebase
Mm-hmm.
consensus, and it runs the genome conf.sh.
Mm-hmm.
I think this script runs called gets the read depth coverage
Mm-hmm.
from the BEM file and gets the total number of bases as well as collects the number of bases that got changed from the VCF file
I see.
and then calculates the QV out of that.
Mm. I see. Okay.
Yep. Okay. Are there any other questions? If not, what time is it? It's past an hour, so I guess if you have to leave, feel free to leave. I can show you a few high-C heat maps. Um, mm, mm, mm. Don't have any good selected examples, so I guess I can just go over some of those. Most of them here, shown here, are not curated, so they might not look so well. But anyway, the caveat here is that if you this is produced by Juicebox with the .hic files taken as an input, which is a conversion of the BEM file, I think, to this interaction for better visualization. So here, each of the boxes, this box here is a scaffold. So if it's the diagonal, means that it's the same scaffold. So you're comparing the read 1 and read 2 alignment. You are looking for where those pairs got aligned and mark that on the diagonal, the coordinates of that. So if you have two reads aligned to the same scaffold, it will be one dot within this box. If it's very close, it will be along the diagonal. This is the beginning, this is the end of the scaffold. So if you see like this big band here in the diagonal, this means that there are many interactions within the short distance in this scaffold. And you can see that it's kind of blurring out, it's gradiating out when it goes far, which means that there are some small interactions that have very long distances. For example, a dot here is an interaction between this position and this position. And so how can we detect some obvious misassemblies? Here is a good example. You expect usually the diagonal to be straight if you see some like this kind of mosaic looking like heat map with a very strong interaction here. That means that this part and this part have stronger interactions compared to this part and this part, right? So this means that this part and this part should get swapped. This is the very basic caveat. So you're looking for things that have this off-diagonal signal, strong off-diagonal signal.
And how do you think that happened? I mean, because IC is exactly that, right? So it should have been detecting that event and use it for scaffolding.
Right. Sometimes it happens when, I think, but most of the cases are now resolved. The Salsa app had, the Salsa software wasn't detecting any misassemblies within a contact before, within a scaffold, I mean. So it's not like aggressively trying to resolve any disassembly that was placed before.
So if there is something generated prior to the last assembly step, scaffolding step, then you inherit it.
Right. That was most of the cases, like either Scaffolding by Nano were doing that, or sometimes you see some disagreements between platforms. KC is kind of, how can I say that, very dependent on the status of how the chromosomes are staying. So you could sometimes see very strong signals for telomere to telomere interactions, right? So sometimes it joins two chromosomes that were localized very closely within a cell.
Yeah, that was my fear in the first place with this data. So.
Yeah, and expecting that might happen to a few of the microchromosomes in the birds, because they don't really have a centromere, I think. They have a different type of centromere, which is scattered all around the microchromosomes, and might have some interactions because of the sequence similarities and so on.
I was wondering, is there any consideration that can be made about read orientation here? So if you were to color the reads by being purplely oriented or not, do you think this would make sense? Or think about how the IC library generation is carried out?
I think it wasn't so easy, but I think it's to.
Yeah, I'll think about that too.
Yeah, I'll think about that. But I only remember that you can't really get the best orientation out of the reads because you lose the orientation information while doing the linking. So sometimes you're not sure whether you're linking this way or the other way, kind of. Or just pulling out whatever got picked, right? So the template itself may not be 100% the same orientation.
Right.
Does that make sense?
Yeah, that's
Yeah, so it's hard to make an assumption that the read pair location are representing the exact same, or that's why high C is prone for inversion error types.
yeah, right.
I just wonder whether the trainee gonna have some possibility that we can try to correct for this error ourselves before we proceed to the next step. I just not sure whether if we leave this error. Well, it doesn't seem to be a problem because the error polishing and the free break seem to be like 5 scale correction. But in general, I think most people prefer to correct this before we proceed with those steps, right? And I feel like it's a little bit uh, cumbersome for Christine if we have to ping her every time we see something problematic for her to come to fix, and we have to wait for her, and then we proceed afterwards. So what's your plan on that?
So what we discovered from the first data release genomes was there wasn't actually that many problematic regions left.
I see. Do you
Yeah,
think
it
it's
was,
because they got scaffold by BioNano first and then high C by that order that make it good, or in general
yeah,
data is good? I'm not sure.
yeah, in general, if you get bigger contigs or scaffolds fit into the next step, you get better scaffolding results, was what we observed.
I see. And
Yeah.
I think in the soon future, I think we're gonna have like ability to see this before the end, right? Because we're gonna add some step to the salsa, those kind of thing. So if we spot this kind of error, what should we do in general? Should we like ping Christine to take a look, or like what should be the course of action?
Yeah, in general, the structural errors are not really affecting the polishing step.
Mm-hmm.
And what Kirsten is gonna do is she's breaking those points, find the break points. If it's an inversion type error, invert the sequence and place it back.
Mm-hmm.
So it's the polishing steps wouldn't necessarily help that much at that point because the breakpoints are gonna be ambiguous anyway.
I see. And
Yeah.
but there's a step that we run the arrow polishing, right? And I heard from actually from your team before I joined VGP that you decide to remove the PBA jelly and use the arrow instead because arrow can fill the gap as well. From what
Right.
I see on my end, I never tried that on the scaffold, but I never see the contig joining by the arrow before. So I just wonder, have you all verified that the arrow can join the scaffold within the contig within scaffold, or it can like basically just to have to concern. First of all, have you verified that the arrow app that we provide you guys can join the contig within scaffold? And second, because there's gonna be some joining going on here, do you concern that if we leave the uh, mid scaffold from the high C and let it run to the arrow polishing, we're gonna have an event that basically mess up the joining of the contig here during the arrow polishing.
So far, I have seen that arrow is basically fixing or filling gaps that are in the size of an indel.
I see.
So for most of the scaffolding gaps, for example, that the gaps that was placed by BioNano,
Uh-huh.
which are like over a meg or something,
I see.
can never get touched.
I see.
So yeah, only the gaps placed by Scaftenex, I think, were mostly targeted. Sometimes BioNano places gaps like a few 10 bases or something like that.
Mm-hmm.
Those kind of gaps might get fixed, but it all depends on the sequencing, on the read alignment.
I see. That makes sense.
If a read crosses that gap, then with more than certain coverage, then the gap gets closed. But if not, it will just remain there.
I see.
Mm-hmm.
Thank you.
And yeah, and because the PacBio reads are relatively short versus the other platforms that we use for scaffolding, we don't expect that the PacBio reads will cover most of the gaps and fill in gaps properly, because the ends are already kind of ambiguous from the PacBio reads point of view, right? It was broken by Falcon already because there were some certain ambiguity to go along.
Okay.
Mhm.
Thank you.
Okay, any other questions?
Sorry, I have so many questions, but did you guys have the pipeline to do the telomere signal as well? And basically,
yeah.
I try to plan like what are the apps that you guys need to develop because we're gonna have training next week, right? So
Yeah,
a good example we can distribute. Yeah.
sure. If you can generate that, I will be very happy. I'm looking for the browser. Where did that go? Where did that go? Did I just close it?
I think it's around it's a little bit late. So if you already have it, it's fine. We can communicate afterward, and then
Yeah.
we can show to the trainee next week who join the app development that they can work on this, or we can help them.
Yeah. Yeah. All the scripts are on the GitHub page, so I will send you later where that is.
Sounds good. Thank you so much.
No problem. So yeah, the telomere script was provided by Sergey, and the Hi-C heat map generation script that runs Juicer inside was mostly provided by Jay. So yeah, I forgot to turn off the noise. Okay, if you have any questions, feel free to throw them into the training channel. If it's a bug on DNA Nexus, there is another channel called App Development. You can post it there. And if it's a general assembly regarding questions, there's another assembly channel. Shane and others are mostly paying attention to that channel, I think. So if you need some more help, you can always throw questions over there. Okay. So I guess I can close the recording. Can I stop that?